Hi, I'm Ushma Neal with the Journal of Clinical Investigation and would like to welcome you to our series, Conversations with Giants in Medicine. I'm honored to be joined today by Dr. Doug Lowy and Dr. John Schiller from the National Cancer Institute in Bethesda. They're in town to accept the 2017 Lasker DeBakey Clinical Medical Research Award. Their combined efforts form the basis of the biology behind the vaccine for HPV, human papillomavirus, which underlies cervical cancers as well as cancers of the throat, anus, vulva, and penis. With widespread uptake, this vaccine has the potential to wipe out these cancers in a generation. We welcome the chance to speak with you both today, and congratulations again on your Lasker Award.
Thank you.
John, let's start with you. Can you tell me a little bit about where you grew up and what you were like as a kid?
Well, I grew up in Madison, Wisconsin. And I was actually quite a rambunctious kid, so very active, but I also read a lot and always had an interest in natural history. And my parents weren't particularly academic. My father had a small business where he sold tractors, John Deere tractors, and my mom was a homemaker. So we had a lot of emphasis put on education, but I certainly didn't have role models in terms of getting into science.
What did you think you wanted to be when you grew up?
Well, initially I thought I wanted to be a park ranger because I really liked the idea of natural history and being out in the woods and things like that. But then I got to realize that really what park rangers do is deal with unruly tourists. And so I pretty much got out of that for a while, so I thought I'd be much more interested in more academic pursuits.
So when did science come into the picture as something that attracted you?
I think I was always interested in science. Initially, again, more natural history. I always studied all the plants and animals and stuff that are in the area and could identify them. So I always had a very scientific bent, I think, ever since I was little. But it wasn't exclusively science. I was interested in many other academic pursuits like history and things like that.
So you went to college with the intent to study molecular biology, or did that come on later?
I think I was one of these people who never wanted to commit, so I didn't have a major until my junior year, but I took all the prerequisites for molecular biology, from the physics to the math and everything else. So on some level I knew I was going to do it, but I just didn't want to make that commitment. But so I think almost from the beginning I knew that's what I was interested in.
So you had stayed in Wisconsin up until this point, but then chose the other W state and went off to Washington for a PhD in microbiology. What made you decide to do a PhD? And why microbiology?
Well, I knew I wanted to be a researcher, and the only way you could be a researcher and make a decision as to what you wanted to do was to get a PhD. And switching from molecular biology to microbiology really was no switch. And the reason I went maybe formally into microbiology is because that's really where the best molecular biology was being done. A lot of it, like in E. coli and some of the phages, the bacterial viruses, where some of the most interesting molecular biology was being done. It seemed a little bit daunting to be trying to understand the molecular biology of higher organisms at the time, given the tools at hand. So it really wasn't a switch.
Okay, well, we'll come back to your trajectory in a moment. Doug, can you tell me a little bit about where you were born and grew up?
Sure. I was born in the Bronx in New York City and grew up there and stayed in the Bronx more or less through high school.
You don't have an accent at all, or can you turn it on?
I may need to come back for a recharge. I've been living outside of New York for quite a long time, down in Bethesda, okay? But my parents were both general practitioners they worked in the Bronx. And I thought from a very early age that I would follow in their footsteps. I actually think of myself much more as being a follower rather than a trailblazer. So being here with the vaccine, et cetera, and being in research is actually something that I did not envision and didn't seriously think about until I was in medical school and beyond.
Wow. All right, so you knew that you were going to do this, but were you a good student?
I was a good student, but I was very interested in sports, electric trains, building model cars and things like that when I grew up. Very little interest in science, more interest in disease and health, because my parents would tell me about the relationship between cigarette smoking and cancer, for example, because that was being identified really when I was growing up as a teenager. And the switch from watching baseball games on TV, where if there was a home run hit, there would be a certain number of cartons of cigarettes that would be given to the local Veterans Administration hospital, to fast forward a few years later when you must not smoke, was really quite a sea change and brought home the importance of prevention to me as a modality.
Interesting. So you knew you were on the path towards medical school, and what made you decide on NYU? Because you just love New York so much?
So let me just say, I followed in my brother's footsteps when I went to college. I followed in my mother's footsteps when I went to medical school, which is, two of the reasons for saying that I really think of myself being more trying to imitate things of people that I respect rather than just sort of striking out on my own.
When you went to medical school and graduated, what made you decide on infectious disease and internal medicine and dermatology as your focus? It was dermatology, my apologies.
Yes, well, what happened was that first I became very interested in infectious diseases as a result of taking microbiology when I was a medical student. I was particularly intrigued by the positive symbiotic relationship between microorganisms and the host. It was not something that I had ever thought about before. I had always thought about microbes as being universally harmful rather than they might be potentially beneficial. And the balance between being inoffensive or neutral, being beneficial and being harmful, was something that seemed really intriguing to me. And the relationship between, if you will, the parasite and the host, whether the parasite was actually causing disease or was beneficial, those interactions just struck me as fascinating and was a major push for me to become interested in doing research.
You did some research during medical school with Jan Wilczek, right?
Yes.
What was that experience like?
Well, that was doing research on interferon, and I did an overly ambitious project, but luckily for me, Dr. Wilczek thought that I had potential, and so it led to my getting a position at the NIH, where I received really extensive training in laboratory-based research, and really got me launched on my research career.
So that was going to be my next question, was how did you end up at the NIH? Did you know that you wanted to pursue academic medicine?
No, the Vietnam War is what got me to the NIH. It was when I graduated from medical school, it was still at the time where the selective service system it was in existence, and recent medical school graduates, if they were male, basically had to do some kind of uniform service activity. Most people ended up going to Vietnam, but you could, if you were selected, go to the NIH for research training, and that's what I ended up doing.
And then you never left.
Essentially.
Let's go back to you starting your career at NIH. What did your laboratory originally set out to study?
Initially, I worked on retroviruses, which is what my training was when I was at first at NIH in the Institute of Allergy and Infectious Diseases. And that initially was replication competent retroviruses. And then it went into the retroviruses that had oncogenes, acute transforming retroviruses, and then moved into papillomaviruses as another version of viruses that could cause cell transformation, but were much less studied than the retroviruses were.
So John, how did you decide to do your postdoc with Doug?
It was actually when I was starting to do some strategic thinking about science. And so my PhD, which was in diphtheria, was quite mediocre, okay? And I was actually a little bit disenchanted with bacterial pathogenesis because it seemed very balkanized. Everybody was studying their little bug, and there didn't seem to be these, overriding common elements that were really interesting. It turns out later on there was, but at this time, and not so much. And this was just at the time that tumor virology was coming up. And I just thought this was so cool that there was these different microbes that were all attacking certain primal nodal points in responsible for cell division, and that would be really cool to study. But my PhD wasn't strong enough, I didn't think, to apply to the really super top labs. And so I went to Fred Hutchinson, where they actually had a very strong tumor virology group there, and I went to every person that was there and asked them the same question: who's young, up and coming in the field, and a really good person? And there was several different names in the groups, but at the top of everybody's list was this guy, okay? So I had to write to him because I asked the right questions. And so what I did is I applied to his laboratory to work on retroviruses, which, as Doug said, that's what he was mostly working at the time. But he wrote back and he said, well, that's fine. What you propose is a nice thing, but I'm just starting to work on these papillomaviruses. And when I read papillomavirus, I really went, papillomaviruses, what are these? I don't know anything about papillomaviruses. And so I read a little bit about the limited knowledge and went, wow, this is really a time to get into papillomaviruses. And so I wrote back with a project related to papillomaviruses, which we actually ended up doing. And Doug, fortunately, who was just starting his lab, had a position available. Naive as I was, I only applied to 2 different positions for postdocs, one here and one at the Rockefeller, one here at the NIH and one at the Rockefeller. So it worked out really well that I got to go to Doug's lab.
I take it he didn't sell himself as doing a mediocre PhD when he applied.
He wrote a terrific proposal, and I was very impressed. And I thought that it might be a good match. And John clearly saw the potential for studying papillomaviruses. Essentially, nothing was known about their molecular and genetic organization. And so we spent a fair amount of time using a model system, the bovine papillomavirus, to work out the molecular genetics. And it was very interesting.
Just so you know, actually he did realize I didn't have that good a PhD because was an NSF grant that we got. He goes, this is really a strong proposal. And I wrote most of the proposal. He goes, the only thing that's a little bit mediocre is your publication record over here, but we still have a chance to get it. And so we ended up getting it. So he was well aware of it.
It was a National Research Service Award from NIH. And in contrast to many other people, we actually conducted the research that was proposed in the application.
John, you tell an interesting story about one of your first aha moments being the second seminar that you went to when you got to the NIH, when Harold Zerhausen came and talked about HPV underlying cervical cancer. Did you then come back to Doug and say, no more BPV, we're studying HPV?
No, actually not. What it made me think is that initially that what we were studying on BPV was just much more significant. And so we continued to study BPV, which is bovine papillomaviruses for people who don't know that. But we started to transition to HPV because we started to realize that there was important differences between the way BPV transformed cells, make them carcinogenic, and the way HPV did. For instance, we looked to see whether BPV interacted with P53 very early on, before it was done with HPV, and we couldn't find any interaction. Until this day, it still does not. But HPV, one of its primary activities is it induces degradation of P53, this critical tumor suppressor. So as we started to realize that all the different types weren't doing it the same way, we sort of transitioned more into studying HPV, which actually was doing things much more subtly than BPV in terms of cellular transformation.
At what point did you start thinking about a vaccine?
We started thinking about that in the early 90s. We had been convinced from the molecular biology that HPV was important for cervical cancer. There were data from epidemiologists that were not totally clear, largely because of false negative and false positive results that implied that maybe papillomaviruses weren't so important. But in the late 80s, it really became crystal clear, largely as a result of molecular biologists developing precise PCR primers that were able unequivocally to establish that the development of cervical cancer almost always was the result of HPV infection. And so there just wasn't any question about it. And we, at the same time, were thinking about the structural proteins of the virus instead of the genes and the proteins that regulate the virus. And quite by accident or fortunately, the first experiments that we ended up doing were experiments that were able to test whether expressing the major protein of the papillomavirus would self-assemble and form virus-like particles. Another thing is that Reinhard Kernbauer, who was the dermatologist, postdoctoral fellow in the lab, was willing to do this high-risk research. But coming back again, we were very fortunate that the first set of experiments where he expressed L1, they made particles. And so at that point, it was a question of asking, well, might this be a vaccine?
In the early 90s, you got this talented dermatology postdoc from Vienna to come and take on this risky thing that nobody else had been able to figure out, how to assemble the coat proteins. So what made you think that the coat proteins of HPV might be an interesting thing to study, perhaps with the potential to become a vaccine?
Well, at that time, it was actually known that if you take, again, mostly in this bovine model, if you take real viruses and denature them and inject it into the muscle of, say, a rabbit, that the antibody, you got a lot of antibodies to that protein, but those antibodies didn't block infection. But if you took the real virus and injected it in the muscle, where incidentally the virus does not reproduce, so it's just the antigen, then you got a lot of antibodies. So we sort of realized, well, it's probably that you need to have the right conformation of this protein. And so how can we get the right conformation without having the DNA, the guts of the virus there? And there was already some indications that some virus types could assemble into virus-like particles. Sometimes in order to get neutralizing antibodies, you needed just the major. Sometimes you needed some other coat proteins as well. And so it wasn't clear that just this one would work, that it would assemble. And for papillomaviruses, they have an unusual life cycle because normally the virus is only made in skin cells, terminally differentiated skin cells, not in normal cells that are replicating. So it was unclear whether there might be chaperones in these differentiated skin cells that are required for assembly. So it wasn't a slam dunk that this was going to work. And we actually, I remember talking to Doug about this, and we put a lot of intellectual hurdles as to why this could, may or may not work. But again, the idea is high risk, high reward. But we with this BPV model, without doing too many experiments, we could figure out whether this was going to work or not. And so we went ahead and tried it.
So there's an interesting anecdote that you guys were puzzling over how to get it to form these virus-like particles. At what point did you have the face slap of understanding or the aha moment that maybe the HPV 16 strain you were using had a mutation that was causing it not to self-assemble correctly?
It didn't work.
It was very difficult. We were really disappointed because when Reinhard did the same thing, expressed it in insect cells, exact same vectors, 16, we could barely find any particles at all. But about the same time as we started 16, we started to make VLPs from a rhesus monkey papillomavirus. And the reason we were going to do this is because we wanted to show that not only rabbits and mice can induce high titers of antibodies that block infection, but that this could also happen in a non-human primate. And this rhesus papillomavirus turns out to actually be very closely related to HPV-16, more closely related to HPV-16 than the next most common type of human papillomaviruses that are in cancer. And when we tried to make the VLPs of that, it worked just as good as BPV. So we thought that it wasn't that there was two classes, one that did assemble and one that didn't. The reason why that was maybe a possibility is because BPV is a cutaneous virus. It replicates in normal type of skin on the cow, and HPV is more mucosal type. And so we thought maybe there was a difference between these two classes. But rhesus papillomavirus, for all the world, is like HPV-16. It even causes cervical cancer in macaques. And so that's really where we really thought, well, the most likely possibility is that the prototype HPV-16, which had been used in most of the labs around the world that Harold Zerhausen had originally isolated, was a mutation because it was isolated from a cancer. And cancers, as we all know, are notoriously genetically unstable.
So let's fast forward a little bit. You started out this pilot trial with the NCI at Hopkins in a small group, and it was amazingly successful. And so you start to try and think maybe some pharmaceutical companies might be interested in taking this up and bringing it past the finish line. First question, do you think that the unique nature of NIH was what allowed you to get to that point in the beginning? And has that been what has underpinned why you both have stayed there for your whole careers?
Well, the freedom of the intramural program at the NIH, certainly for John and me, was critically important, in large part because we had never worked in this area before. We had studied the genes that were responsible for transformation or regulating gene expression, etc., but we had never studied the structure, had no background in immunology or in terms of vaccinology or things like that. And one of the big advantages of the intramural program is that you sort of have level resources that you retrospectively defend how you've used them. And having somebody like Reinhard made it straightforward to do those pilot experiments, which once they bore fruit, it was clear this was something that we could try to pursue. On the other hand, it's not as though it was impossible. There actually were other labs that were trying to do things along the same lines. In retrospect, I think we were fortunate that because we had worked on BPV, that's where we did things first, and we were able to show not only that we could get self-assembly of particles, but very quickly, because we had developed a neutralization assay in vitro, in a very short time, say, very high levels of neutralizing antibodies, much higher than with most other vaccine types. And that then gave us, if you will, the positive control to then be encouraged to go forward with the human papillomavirus studies. Other people who were working in this area tended to focus on HPV first, for which there either was no neutralization assay, or one of the HPV types, there was a neutralization assay, but it was very cumbersome. And for example, the results were published a couple of years after demonstrating making virus particles.
Can you talk to me a little bit about why you thought pharmaceutical companies would be interested in this? How many cervical cancer cases are there a year?
Well, there's about 500,000, 500,000 worldwide, and about 250,000 deaths. But from the point of view of the pharmaceutical industries, I think their biggest issue going into this, which was a reasonable one, is that the track record for STI vaccines at that point were miserable. You know, people had tried to do herpes virus vaccines that seemed to work in animals and they didn't work in people. Chlamydia, people had been working on some HIV stuff, and none of them seemed very promising. And I think sort of the biggest intellectual hurdle was, it's not like if a flu vaccine where you may be exposed once or twice. If you have a sexual partner, you may be getting exposed repeatedly, over a long course, over a course of years potentially, because these viruses tend to persist. And so the idea is that people couldn't get their head around was that we could induce long-term sterilizing immunity. The idea that, if you delay infection by 6 months, so a woman gets cervical cancer at 50 in 6 months versus at 50, from a public health point of view, it doesn't make a whole lot of difference. And so that was very difficult for them to get their head around. And so it was really kind of a leap of faith for Merck and GSK to spend hundreds of millions of dollars to test our vaccine and see whether they would work. I mean, again, to bring back the HIV, about the same time as they were doing the HPV vaccine, like for instance, Merck did an HIV vaccine trial, which the HPV worked great, spent hundreds of millions of dollars to show that it works. The HIV, they spent hundreds of millions of dollars and they got actually nothing out of it other than know that their particular vaccine doesn't work in people.
So speaking of that, I mean, this was your baby. You had seen some initial positive data. Did you know that it was going to be as successful as it was when you were watching the Merck and the MedImmune/GSK trials?
Certainly not immediately. And for us finding out about the results of the monovalent HPV-16 trial that Merck conducted and that was published in 2002, but it was first announced in 2001 at a papillomavirus meeting, was simply astounding because there were 41 cases of HPV-16 infection in this clinical trial. All 41 cases were in the placebo group and 0 in the HPV-16 group. For us, this clearly indicated this was going to work and be highly effective because you just don't get results like that ordinarily in a clinical trial.
Did you have some champagne?
Actually, we weren't together. I was. It was at a meeting in Brazil. Doug usually goes to, it was at the International Papillomavirus meeting, which is usually about every year and a half. And I was actually the moderator of the session. And what was interesting is the person who did it, Laura Kautsky from the University of Washington, who was the lead epidemiologist on the study, didn't tell anybody that she was going to talk about it. And so we were all sort of dumbfounded when it was, and I remember afterwards saying, ladies and gentlemen, you've just seen history.
Okay, so I'll change my question. Did you have caipirinha?
I had a caipirinha afterwards. Actually, I do remember that. Absolutely.
Right on. Well, it must be tremendously gratifying. Can you give me the data on how many doses of the vaccine you think have been given so far?
It's about 50 million. It depends on whether you look at it at the full schedule or at least one dose. And it's maybe 60 million with one dose. And I think the important number is IARC, the cancer branch of WHO recently came out and said already, even though we have very little reasonably small uptake in the developing world, is that approximately 400,000 of future cervical cancer cases or deaths have been averted by vaccination so far, and we're really only getting started.
So let's talk a little bit about uptake of the vaccine. First, let's talk about the developed world. Is it a little bit frustrating that there has not been complete uptake in places like the United States?
We had expected that uptake would be greater than it has been, but I think that we're going to see continuing increase. Every year now, for the last few years, there is a substantial increase in the percentage of boys who are being vaccinated. The increase in girls is less than boys. But I think we're gradually getting to the point where probably in a couple of years, it will be at the 65 to 70% level, both for boys and girls. This is enough to really be starting to see real herd immunity. The countries where there has been high uptake, there's been dramatic reduction in HPV infections and the short-term disease. The problem, of course, is that preventing cervical cancer and the other HPV-associated cancers will take 20 years or much longer than that because of the long interval between the infection and the development of the cancer. In addition, the real big public health area and the need is in the developing world, and we're barely scratching the surface there. It's one of the reasons why I'm very excited about a new clinical trial that we are just embarking on to test whether one dose for young adolescents might be sufficient to confer long-term protection. Because if that were the case, I really think it would become feasible to try to vaccinate the whole world.
Let's pivot a little bit. Doug, you have been Deputy Director of the NCI since the 90s, and for the last 2.5 years, the Acting Director of the NCI. Your research has been so fascinating. We've spent the last 40 minutes talking about it, and I can see your real excitement. So what made you take on more administrative roles? And what have you learned from them, if anything?
I felt that I had gotten a tremendous amount from being in the intramural program at NCI, and I wanted to see whether I might be able to do more, to some degree really, as a way of giving back. And in the 90s, I started out as Deputy Director of the intramural NCI program, and I did that up until 2010. And when Harold Varmus became the NCI Director, he asked if I would become the Deputy NCI Director. And before I said yes or no, I turned to the man on my left and I said, "John, should I do this?" And he said, "Yes, you should." And and I asked the person who was the director of the intramural program, and he thought I should. And then I asked the most important person, and my wife said I should. So I ended up doing it. And then when Harold left, I became the acting director. And it has been very interesting. And you know, part of what I love about doing research is being able to address some important questions. Many of them are basic. Some of them have translational and clinical implications. But being involved at the policy level gives you an opportunity to do more, although not as directly. You can help researchers with the development of their career. You can try to put more emphasis in one area of research as opposed to another. And there are just many different ways in which you can try to help the cancer research enterprise. And a very important part of it for me has been to try to strongly support investigator-initiated research. Because to me, that is a very important part for the breakthrough findings of today that then lead to the clinically important interventions of tomorrow.
Just so you know, this guy is a more outstanding scientist and at the same time administrator than anybody I've ever met. Okay, he can juggle more balls in the air at the same time than anybody and do a really great job for all of them. Now, I personally have not gotten involved in administration because I think the NCI wouldn't be getting good value for their money, because I'm a reasonably good scientist and I would be a really mediocre administrator. So I've sort of learned the Peter principle, and so I stay in science where I probably
John,
can
you're
do the
very
most.
self-aware. Do you sleep?
Do I sleep? Yes.
But I lots of times get emails from him at 2 and 3 o'clock in the morning, so just so you know that.
Have you gotten to meet either president under whom you have served?
Well, actually, we both met President Obama when we received the National Medal of Innovation and Technology, and that was certainly a very exciting time. And then last year, because of the Cancer Moonshot, I participated in a meeting with President Obama, Vice President Biden, and a number of the cabinet secretaries that were focused largely on cancer research. And that was an extraordinary experience.
If you could not have been a doctor or a scientist, what do you think you would have done in your lives? What would have kept you engaged?
Well, if I had the talent, my first two choices would either to have been a wonderful miler or 1,500 meter runner. Second choice would have been to play football in the NFL. But when you're talking about what might have been feasible, I would have really liked to have been a high school biology teacher.
How about you?
Well, along Doug's lines, maybe a professional trout fly fisherman, but you know, it's getting me a little bit old for me to do that. But I guess I was always really an academic, and I think I realized that that really was the perfect thing for me. So if I couldn't do science in sort of an academic setting, I probably would have been something like a historian, you know, could sort of study the flow of ideas and cultures and when cultures interact and things, you know, things of that nature.
Do you guys ever fight?
No.
Have you ever heard each other yell?
No. We've heard each other yell. Yeah, not at each other, but maybe I've heard Doug yell in 30 years or something once or twice. I'm not sure you've ever heard me yell. You've seen me get mad, but
Yes,
not,
yes, yeah.
but not yell.
So I think this is really the story of partnership and collaboration and what you can do when you work together. So thank you both so much for joining me today. It's been a real pleasure hearing a little bit more about the two of you and the men behind the discovery. So thank you so much.
My pleasure.
Thank you.